Super, right. So I was here 20 odd years ago for the first CTC meeting, and still some people in the audience who are the same. So it's great to see everyone. So I'm going to talk today about work that was done by Lele Kui, who's in the photo. We're going to talk about the phenomenon of dominance in mammals. And surprisingly, he, I should say, found quite a lot and found some quite interesting stories underlying this. So, what do I? Is it the keyboard to move? Oh, there we go. Okay. So very briefly, what do we mean by dominance? So normally when we do a genome-wide association study, we assume additivity. So that means that the phenotype of the heterozygote is going to be halfway between the two homozygotes. But there are various types of dominance. There's partial dominance where the heterozygote is sort of still within the range of the two homozygotes. Or there's complete dominance, which is kind of the Mendelian idea of dominance, where the heterozygote is going to be exactly equal to one of the two homozygotes. And then there's overdominance, where the heterozygote is outside of the range of the two. And this is obviously the most extreme type. But we're going to use the word dominance to mean all these three types. And the way we look for dominance is with a fairly straightforward extension of the standard linear mixed model. So we code up our genotypes as both additive dosages. So genotypes are coded as 0, 1, or 2. It's just a number of reference alleles. And then we also have an additional coding, which is just a 1 if the individual has a heterozygote dosage. And then you can model any type of these different sorts of dominance by a linear model of this form, X times some regression coefficient beta A plus D times the regression coefficient beta D. So you just choose beta A and beta D appropriately. And then the error term here we model as a combination of both the additive and the dominant kinship matrices, which are computed from these additive and dominant dosages. So this is all pretty standard. I'm not going to go into details. So we're going to look at three cohorts of animals. These are all published data sets. The F2 pigs are from Lele's university in China. And so one thing to know about pigs is they don't form inbred strains. So this is a cross between a Chinese and a European breeds of pigs. And if you're interested in history, the pig was domesticated independently somewhere in the Middle East and somewhere in China. And so they are quite genetically diverse, although some Chinese pig breeds were brought over to the West a few hundred years ago. So there are now a few Chinese haplotypes floating around in European breeds. And the European pigs have been selected to be lean, and Chinese pigs have been selected to be fat, okay, because the meat tastes better. So yeah, in a lot of interesting genetics. The other two populations are the HS rats. This is Amelie's data set from 2011, I think, if I got the year right. And then the HS mice, which is from even earlier, from Will Valdar and Jonathan Flint. So we have basically 1,000 or 2,000 animals in each cohort. We have hundreds of phenotypes, and we have several hundred animals measured for gene expression as well in various tissues. So we can estimate the additive and dominant variance components for heritability in the three stocks. So these are SNP-based heritability estimates, I should say, just using those genetic relationship matrices that we used in the linear mixed model. And you can see that the total amount of heritability is given by the sort of the height of the blue, and then the amount which is dominant is shown in red. And as you would expect, the majority of traits are predominantly additive, but there are significant amounts of dominance heritability really across the board. And something like 25% of the heritability appears to be dominance, according to this measure. So, because I haven't got much time, I'm only going to kind of give you a taster of all of the results we have. So I'm just here showing you the results of heritability in the F2 pigs. And on the right-hand side, we have the average additive and dominance heritabilities for different types of trait. And you can see for some traits, the amount of dominance is really quite large. For others, it's quite small. And we have the usual missing heritability problem that we can map only a subset of the, we can explain only a subset of the heritability using QTLs that pass genome-wide significance. We mapped lots and lots of QTLs, and here I'm showing you the results for all three stocks. The figure's gone a bit weird. Apologies for that. I think this is transferring to a Windows computer from a Mac. So we've color-coded QTLs according to the amount of dominance. So basically red means they're dominant in some way, blue means they're additive. And you can kind of see there's a wide spread. There are also lots of QTLs concentrated in particular regions. These are things like the MHC, for example. So here we're looking in the pigs, and I want you to focus on the right-hand figure here. So what we're showing here on the x-axis is the difference in log p, that's the measure of significance, when you fit what we call the AD model, this additive dominance model, versus if you just fitted an additive, standard additive model. So you'll notice that most of this axis is positive. So most of the time we see an improvement in statistical significance when we allow for dominance. And we can map a significant number of additional QTL by doing this, even though there's a penalty in degrees of freedom if it's an extra parameter, but it seems on average you do better. So, and across all the three stocks, we mapped about 44% more QTLs than just doing a standard additive GWAS. Yep. No, these are physiological QTLs. I'm coming on to gene expression. And most of these additional QTLs, as you would expect, exhibit the more extreme types of dominance. So they're complete dominant or over-dominant. So here's an example. This is in pigs. We're looking at hemoglobin levels. On the top, you have the GWAS that you'd see if you just fitted a standard additive mixed model. And we have this sort of rectangle here over the region of interest, and there's a little zoom in over here. When we fit the dominance model, okay, we get much more statistical significance. Now we're moving on to gene expression. So interestingly, we see much more dominance heritability in gene expression traits than in physiological traits. Now this is completely bizarre because, as we all know, most cis QTLs are additive. Okay, but I'll explain what I think is going on. So apart from in the pigs, certainly in the mouse and rats, we see more dominance heritability. If we sort of position our eQTLs across the genome, so here we have the SNP position on the x-axis and the position of the gene on the on the y-axis, and here we're looking in the HS rats, in 2 different tissues. So you can see the diagonal line of cis-EQTLs. And what we've done here is we've partitioned our EQTLs into 2 types. There are what we call generalized additive. That's basically additive and partial dominant, because that's almost additive. And then generalized dominance QTLs, which are the complete and over-dominant things. So if you do that filter, basically the diagonal bands disappear completely, almost completely, just a little shadow of one down there. So what this is showing you is that most of the cis-EQTLs are indeed additive. However, the trans-EQTLs are enriched for dominance. It's not true to say that all trans-EQTLs are dominant, okay, but they are enriched for dominance. And obviously, we don't have, with only a few hundred animals, that much statistical power, so we can't say for certain that every single dot on this image is a genuine, true positive. But they can't all be wrong. And if we do a test for enrichment for dominance amongst trans-EQTLs, we get ridiculously significant Fisher-exact test p-values. I think it took us ages to actually get R to print out a p-value, which wasn't not a number. Okay, the most interesting thing we found was due to an accidental mistake, where in the HS rat data we have RNA-seq data, and Lele accidentally analyzed the data using transcripts, alternate transcripts, rather than just overall gene expression levels. And by doing that, we actually uncovered some really interesting phenomena, and they're quite common. So what we have here on the top, on this top thing, this is for a gene called FOXJ2, and this has 2 isoforms. If we just do a standard EQTL scan for FOXJ2 whole gene expression, we see nothing. Okay. If we take the 2 isoforms, which we kind of call FOXJ2A and FOXJ2B, we get these humongous EQTLs, which are basically complementary. And if we plot gene expression, or gene expression on the y-axis and FOXJ2B on the x-axis, and the same J2A over here, what you can see is you get essentially a kind of dominance type effect of genotype on expression of these two things. And when you plot the 2 isoforms expression against each other, you get this complementary expression. So the sum of the 2 transcripts expression is constant, but they're going up and down. I mean, we see this for many genes. So, and we've color coded the genotypes here, so you can kind of see how our SNP genotype influences expression. So in this case, if you're an AA homozygote, basically you get 0 expression of FOXJ2A, and it's all J2B. Oops. Oh, I think it's meant to do that. Okay. So if you then classify genes according to how many alternate isoforms they actually have, then, well, basically, the majority of genes in this annotation of the rat genome, which was version 3 point something, 3/4 only have 1 isoform, so you obviously aren't going to be able to see anything. About 17% have 2 isoforms and 6% have 3. If you look for the enrichment of dominance with how many isoforms you have, we get significant enrichment, particularly in the amygdala. So in conclusion, about 25% of heritability seems to be due to dominance across these three populations. There are dominance effects at over half of the QTLs that we measure. Dominance EQTLs are most likely transacting, but cis EQTLs are mostly additive. Alternately, spliced genes are enriched for dominance effects. And I think one thing you should note here, of course, is that these study populations all have high allele frequencies compared to human populations. And you need high allele frequencies in order to get all three genotype classes represented in enough animals or enough individuals that you can actually detect dominance. So the problem we face in human studies is this huge tail of low frequency alleles, which makes it much harder to detect dominance in humans. Okay, with that, I will stop and take questions.